Hello students, today we will describe a topic fertilization of chapter 3rd Human Reproductive System for class 12. So what is fertilization? The fusion of male and female gametes, that is called fertilization. Fertilization is a process that ends with syngamy. Now the discharge of semen by the penis into the female's vagina close to the cervix during coitus is called insemination. How many steps are followed by a fertilization? First step, emission, it means ejaculation. Insemination, okay, release of sperm into female's vagina. Now egulation and capitation, it means sperm moves through the cervix, enter the uterus and reach the junction of the isthmus and ampullae of the fallopian tube. That is called as the egulation and capitation. So that is the second step is capitations, means preparation of spermatozoa for fertilization in female reproductive tract. Now all copulation do not lead to fertilization in pregnancy. The process of fusion of sperm with an ovum is called fertilization. Understood? Now anti-fertilizing chemical of mature spermatozoa attracted by fertilizing chemicals of mature ovum. Understood? Now where is the site of fertilization? Junction of ampullae isthmus, that is the site of fertilization of female tract. Understand? Now how can you development of embryogenesis? We will see this flowchart. That is very easy. In my previous videos we have discussed about this, but today we will repeat. Sperm produced from male, ova produced from female. The fusion of male sperm and ova formed the zygote. One day zygote under the cleavage of holoblastic cleavage, the zygote change into two cell. Zygote is diploid. Sperm and ova is haploid. And two cell stage day for a second day. Now under cleavage, second cell formed a 4 cell stage for a day third. Now 4 cell stage change into 8 cell stage and day for a day third, and 16 cell stage for a day fourth. 8 and 16 cell, that is called morula. Understood? 32 cell stage 32 formed by 16 under cleavage day 15. Now 64 cell formed from 32 under holoblastic cleavage. This process, what will be made? We will made a blastula. Now at the end of the day 5th, okay, or beginning of day 6th, okay, so 6th or 7th day in implantation begins. Okay, implantation means that attachment of embryo to endometrium layer of uterus at the end of the first week. Understand? Now when embryo completely submerged with endometrium lining at the end of second week. Now that is very important, HCG hormones, HCG synthesize secretion at the end of first week. Significant amount production at the end of second week. Now, presence of HCG in urine is confirmatory test for pregnancy, but it can be detected only after the second week of pregnancy. Okay, so pregnancy kit have antibody against HCG. Okay, presence of HCG detect pregnancy after second week. Okay, so HCG is a human chronic gonadotropic hormones. Now, and LH is also, their two hormones are the maintainers of corpus luteum only for first trimesters. Understand that? Now, how can you understand? How can you understand these flowchart? You will see this diagram. Okay, what is this? This is the fallopian tube. Okay, fertilization is here. Here the junction of isthmus and ampullae, and the sperms enter from this extranogenital organs. It means vagina. Sperms enter from the vagina to fallopian tube, and the egg comes from ovary from ovulation period. Now the fusion of male sperm and sperm fuse with ova. Okay. Now what will be formed? They form the zygote. Zygote, under a cleavage there are two blastomers, two celled, and the formate four celled, formate from two celled. Now 8 celled, 4 से 8 and 8 से 16. 16 के बाद आपका 632 and blastomers are formed, and these morula के बाद blastula attach with uterine valve. Okay, now development of embryos. Clear here? Now embryos change into fetus. Okay, now how can you explain with this changes? First of all, zygote, they are diploid. First cleavage after 30 years of fertilization. Okay, now they change into second blastomers. Now next is second cleavage, holoblastic, after 60 hours of fertilization. Change to blastomers, change into 4 blastomers. What is the meaning of ZP and CR? Zona pellucida is surrounded by secondary oocyte and corona radiata surrounded by the eggs. Okay. Now 4 blastomers change into transverse unequal holoblastic. There are two types of holoblastic: equal and unequal. So third cleavage is unequal holoblastic. Look that diagram. Okay, unequal holoblastic change into undergoes compactations, corona radiata degenerate, peripheral cells become flat and leaves no gap junction that has tight junction. This stage is called morula. That is very important stage of morula. Okay, now the next is fourth stage. Fifth is blastocyst stage. Morula, it means inner cell mass. That is where the morula continue to divide and transform into blastocyst. You can say blastocele. It moves further into uterus. This process of transmotion is called blastulation. Blastomers, inner cells, and sticking nutritive trophoblast. This is a trophoblast cell. This is inner cell. Now they change into this silt or pore formed in zona pellucida, while blastocyst emerges out. Blastocyst emerges out from this blastula stage. Okay. तो blastula formed. Now blastula and before implantation. During emergence, sometimes blastocysts break into two parts, separately implant in uterus in the form of identical. अब हमारा ये ब्लास्टुला से फिर क्या इम्प्लांटेशन होगा, ये क्या होगा, यहाँ पर यूट्रस से जुड़ेगा. ये क्या चीज है? एपीब्लास्ट, एमनियोटिक फ्लूइड. अपर लेयर इज हाइपोब्लास्ट एंड दैट इज प्राइमरी यॉक सैक. Inner layers is cytotrophoblast. They are divided to syncytophoblast, cytotrophoblast, syncytotrophoblast. Understood that? Now they are very important cleavage. They are divided into two parts during a cyto or syncytio. Now during changes 7th day from fertilization, blastocysts implanted by sticky trophoblast at inner. They form it ectoderm, embryozonal node, epiblast, mesoderm and endoderm also. Okay, they are formed the 3 layers. Okay, now do you know it very well? Placenta is very important. There are so many changes of these changes in zygotes and changes during a pregnancy and changes in embryos. So that is the main important functions. The placenta is connected to the embryo through the umbilical cord, which helps in transport of substances to and from the embryos. Placenta act as an endocrine tissue and produce several hormones like HCG, HPL, estrogen and progesterone. Now that is very important. 3 layers are formed. It means a gestulation period start. After implantation, the inner cell mass differentiate into an outer layer that is called ectoderm. Inner is endoderm and mesoderm is soon developed between the ectoderm and endoderm. So thus transforming the blastocyst into a 3-layer structure called a gastrula. By process is called gastrulation. That is a gastrula. Gastrula में 3 layers बन जाती हैं. The 3 layers give rise to all tissue and organ in adult. Inner cell mass contains certain cell called stem cell. That is very important, stem cell, which have the potency to give rise to all the tissues and organs. Understand? Now changes in extrachromatic membrane. That is very important. Changes. Now do you know extraembryonic membranes, amniotic fluid? This is the embryo and amniotic fluid is formed between further ectoderm and trophoblast. After morphogenic movement, yeah, changes in embryo. Okay, the morphogenic movement are seen due to gastrulation. gastrulation के बाद हम बहुत सारे changes देखेंगे. क्या क्या changes? एपिबोली, इम्पोली, ingression, invagination, involution, etc. Now they are formed the different types of layers. Amnion. Do you know embryo? The covering of embryo, the amnion. Okay, that is has outer mesodermal lining and inner ectoderm lining. Amnion is a protective layer. Amnion is important. Amniotic fluid has worn out and dead cells or waste cells of embryo. When it is taken for diagnosis, then genetic abnormalities will be identified. Aminocentesis. This spelling is amniocentesis. This is the right spell. This is related to amnion. Understand? Publicly, amniocentesis is banned for fetal sex determination. That is very important. Embryo किससे covered होता है? Amnion से. This is amnion and this is amniocentesis process. Chorion lined by ectoderm external, that is lined by ectoderm external, mesoderm internally. It helps in diffusion and respiration. The example respiration, बेबी को सांस किससे? कोरियोनिक से. And allantoin has outer ectoderm and inner endoderm. It is considered as urinary bladder of embryo. That is work as a urinary bladder of embryo. Okay. Yolk sac, that is very important. Mesoderm is outer and endoderm is inner yolk sac. Okay. Now in human yolk sac has no yolk. Yolk sac placenta is present in shark. Now this is chorionic villi. Okay. Do you know the stems? Next is